systemized knowledge by observation and experimentation in order to determine the nature or principle of whatever you're studying. That's a whole mouthful, but when push comes to shove, eventually what we want to do is increase knowledge and establish the facts of the physical world around us, nature, humanity, etc. And there's two very important parts to the science, to making science progress. One is the scientific method, and obviously, if whatever you discover you don't communicate to the rest of the scientific community, your research is of no value whatsoever. So communicating results is extremely important. And that pertains directly to what we will discuss next, which is journal claims. So science is not more than trained and organized common sense. That's what Thomas Huxley already said over a century and a half ago. It's a logical way of thinking and decision-making. Process of systematic observation and systematic testing of ideas. And here's that systematic approach. Usually science starts with things that you've observed. Usually there's observations that you have or background information that you have, no matter where it comes from, whether it's from your own observations or from somebody else's. And based on that information, you formulate a next question. We want to take. We want to advance science, of course, right? And usually, asking a question leads to formulation of a hypothesis. This sounds all very dry, but you're going to be taught in how to put this into practice in year one, in year two. You test the hypothesis by experimentation and by making further observations, of course, right? And eventually, it is the systematic analysis of all this information that will lead you to conclusions, to interpretation of these data, and will bring you back to your hypothesis and reconsider it. So very schematically, basically it's observations, research questions, hypotheses, leads to prediction, and the prediction then tells you what type of experiments you should do. Is it visible from up there? I guess so. Liam?
They are more students than I expected, so I'm telling you. Counting.
Oh, good. You're doing a head count. Excellent. Excellent. Maybe we should have hands raised from those who take the course in Belgium, and we can continue on. Who come from Belgium? All right, Leo, count them. Predictions, keep your hands up, huh? Keep your hands up. Predictions, hypotheses lead to predictions, and that allows you to design your experiments, and they either refute or confirm your hypothesis. All right, you got them?
Yeah.
All right, hands down. All right. So, observations, where do they come from? Observations, I mean, usually they're preliminary data from your lab. Just imagine you're writing a grant, huh? I mean, you're usually writing that because you've discovered something, and we call that preliminary data. Or they come from flyer experiments. Flyer experiments are typically experiments in the realm of, you know what? I think this is a good idea. This is a good experiment to do. Let's just try it. That's a flyer experiment. Serendipity, you were looking for something completely different and you discovered something else. Toeval, in good English. Your experience, of course, counts in all that you do. You have a whole lab journal full of observations, and sometimes these match. And observations or preliminary data come from. Observations, data come from, findings come from scientific literature. Very important. Then, research questions. They define a problem. Cancer kills people. That's a problem, right? But that's very a-specific. How does the cancer kill people? That's already a lot more specific. What mutations play a role in lung cancer? That's even more specific. And so you get a sense that it is extremely important to come up with the right questions. Actually, I think it was Einstein who said, and it's only half on this screen, I have no idea why. Anyways, the formulation of a problem is often more essential than its solution, meaning that it's extremely hard to formulate the right questions in science that help you direct your research. Okay? Most of the times, the objective of a study is clear. Like I just said, if you want to discover what mutation causes lung cancer, the objective is clear. It's not so easy to carry it out. But most of the time, there's considerable background information that must be understood first to frame the problem. And that's what we were talking about if we're talking about literature reviews. Okay? All right. A hypothesis basically is not much more than a statement. It proposes an explanation for your research question. Okay? It can be tested. That's very important, because that's what we do in science. And a hypothesis makes predictions. And eventually, the predictions help you design your experiments to test the hypothesis. Essential steps in experimental design are, in doing experiments are experimental design, carry it out, collect data, and interpret and conclude what you've observed, about what you've observed. And then there's another very philosophical statement here. In essence, the scientific method never allows us to conclusively prove a hypothesis for the simple reason that a hypothesis are usually designed to, the method is usually designed to disprove a hypothesis, right? Experiments can only support or dispute a hypothesis. You can never prove yourself right, because there's usually much more to figure out. You can prove yourself wrong. And then the flip side of that, of course, is if you can't prove yourself wrong, you're probably right. So then you go on again, eh? All right, communicating results. I promised I was going to keep this short. I realize I'm racing through it a little bit, but all of this will become much more clear as you're doing this course. Communicating results happens in many different ways. Lab journals, that's the first way of communicating to your boss, to your supervisor. You write things up. It's an official record of the experimentation that you've actually carried out. There's scientific meetings. Those are actually where scientists come together on particular topics, and those aim to stimulate and accelerate research based on observations that all sorts of scientists make from different angles, approaching the problem, or doing similar experiments but getting different readouts in different systems, that sort of thing. It's all very useful to go to and to help science ahead. The internet, right? You can get a lot of data from internet. For instance, you need to clone a gene, or you need to mutate a gene, and you want to know where to put the mutation. Well, fortunately, most of the human and mouse genes, as a model, are known, are sequenced. So the internet is a good place to get that sort of information. If there's any questions, I expect hands in the air, and you can ask. And then the most used information, the source of information that we use are articles, publications. There are millions, gazillions of articles out there. Many of them which are not relevant to your topic, but a whole bunch which are. And they report on data, on analysis, on interpretation. These basically are manuscripts, right? Goals of publication are to establish a permanent, cumulative record of knowledge, to share results, common research interest built on knowledge, we talked about here, to introduce also novel methods. Some smart cookie may have advanced at one point in time doing science tremendously. A nice example is when I started doing science as a PhD student in America, if you wanted to know about gene expression, you had to run a northern blot. Who knows what a northern blot is? I'm not going to ask you to explain it. All right. Who knows what PCR is? Who knows what a microarray is? See? More and more hands. Methods improve. From northern blot, which was painstaking and annoying, because 3 out of 4 northern blots failed miserably, usually. Then somebody discovered PCR and got the Nobel Prize for it, because we were all so happy that now we had a much more quick and reliable method to test for gene expression. And then somebody developed microarrays, and now we could test 25,000 genes at the same time, expression-wise, right? So, I mean, that sort of things are important as well in publication. Of course, there's always the part where you, as a scientist, gain credits for the finding. You're the one who did it, so chapeau. But at the same time, you're going to be held accountable for your findings, because if the rest of the world cannot reproduce your findings, then either there's something wrong with the rest of the world, or your research wasn't quite what it was supposed to be. And, of course, last but not least, but equally important, publishing results means that somebody else doesn't have to do it. There's always, of course, the replication. People need to be able to replicate your data, your findings, your methods. But there's an end to that, right? If somebody has done it, has been done, then it helps prevent repeating, wasting money, wasting time, wasting energy. All right. So, what we're going to do throughout this block is look at publications that are focusing on topics that are discussed in these modules. So this is done in the form of journal clubs, and one of the most important rule number 1 of journal clubs is that you read the paper. Rule number 2, 1 read. Read all of the paper. There's no such thing as sort of glancing over a paper and hoping that you understand. Okay, what we're going to ask of you, rule number 2, is look at all the data and help me as a tutor and all the other tutors throughout these blocks to see that you understand what these people were going on about in literature. So you have to understand the details of the research. You have to read the paper, be critical. Use the scientific method to dissect papers, okay? What were their initial observations? And how did they get to their specific research question? And do they have a hypothesis? Did they state it actually, or can you come up with one? And then carefully analyze the data. The methodology used, was that valid in terms of communicating to the hypothesis, right? Are the experiments done appropriately? Are there any data missing? Are the appropriate controls present, and are the data consistent with other data that were published? It's not a bad idea to keep these slides next to handy when you're looking at these papers for journal clubs. Do not read the author's conclusions and assume they were right, but make your own, draw your own conclusions. Are there any alternative explanations for the data? Be critical. All right? Have all the key experiments been done? Is there something obvious missing? Sometimes you'll see that. Depending on where you publish a paper, you guys are probably familiar with impact factors, eh? Every journal has an impact factor, meaning that whatever you publish in there literally has an impact. And the more people that read your data, the more impact your data will have. Now, the simple sort of rule is that the better the journals, the higher the impact of your paper will be. So there's Science, there's Nature. You're familiar with these journals, right? And there's Cell, huge impact factors. New England Medical Journal, JCI, Journal of Clinical Investigation, that sort of thing. Extremely high impact, and so usually in those type of papers you will see that reviewers ask for the most nitty-gritty details that they can think of in order to nail your statement, in order to have you convince them that your hypothesis was proven or disproven, whatever the case. The lower the impact of a journal, probably the more crucial experiments are missing. So we have papers from all over the spectrum of journals. And so it's your task to critically look at all these data and tell us if you're satisfied or not. All right? The way we do these journal clubs is by having you guys read all the papers and being able to introduce background hypotheses and predictions if there are any. Explain the figures in the paper, what are the experiments done, and how are they done, and discuss these and being able to discuss these in detail. And then at the very end, you have to be able to summarize and conclude on the significance of the paper. Like I said, be critical and figure out what was good and what was not so good, in your opinion. Presentation of a figure, the way I do it in my module, but I will introduce that in some detail when we get there, is I'll have all you guys prepare the paper and I'll give you turns in the module at the journal club in discussing papers. So you don't know up front which figure you're going to discuss. All right? Which means that you have to prepare together and you have to know what you're talking about. So basically, if you present a figure, what you do is describe the purpose of the experiment, and you stop, you finalize basically by stating, what is the next logical question then, and that's sort of a bridge to the next figure. Describe the methodology, the results in detail. Try to understand all aspects of the figure. If a Western blot contains smudges, why is that? Do we like that or not? What are the conclusions of the authors, and what are your own, and do they differ? And is there, like I said, anything missing in terms of data? And eventually, you'll have to let us know what your conclusion is and what we have learned. I think I'll leave this to Leo. This is on allium. Leo knows a lot more about it, and Theo too than I do. So this was sort of the short version, the bird's eye view on journal clubs. Like I said, every module will be introduced specifically on what is expected of you, and also in terms of what you need to do for these journal clubs. Practical training will, the first one will follow in the junior period, so that's the third block. You're going to try it yourselves. You're going to wreak havoc in the laboratory. We are here now. Here are the two practical training periods, the junior and the senior. I touched upon that already when I started off. And what we typically see is that people want to go broad. There's nothing wrong with that. I should rephrase this. It's very good if you go broad. You learn a lot. Keep in mind that you have to organize this. All right, for the junior, we see that people go abroad, and you'll have to organize this in terms of discussing with somebody outside the university, somebody outside the country, in terms of topics, projects, but you also have to get some money from somewhere. There's another one. What country is this? My, we have a lot of teaching. It's going to be a really sit somewhere down the line. So, in order to figure out what you want to do in terms of practical training, talk to people. Talk to older students, those who are further in their curriculum already. If you end up in a lab or you see teachers that present interesting topics, tackle them. hold them to the ground and ask them, you know, where can I go for cool training periods here? Because you have to figure out. Oh, sorry. You'll be given a digital database where we have an awful lot of projects, both for the junior and for the senior practical trainings. And that's one thing. You'll be sent out for student interviews, and you'll choose your PI, where to do your practical training. We'll ask you to help us organize this, which means that we're going to ask you to include your deadline, uploads and that sort of thing for the junior practical training. Is this correct? The deadline is December. For the senior, I know for sure, April and May. There will be an introduction on the senior late block 1.2, because there's a lot of organization and communication involved with that. So we'll give you a separate introduction on that topic. But one of the things that I wanted to stress is if you want to go abroad, you need to start organizing things early on. Because usually that means that you have to get some money somewhere. Oh, I forgot to say that. If you come here to a lecture, turn off your mobile devices. I heard something whistle at me. Usually I don't object, but this sort of whistles I recognize. Those were not meant for me. So turn your things off, okay? All right. There's many institutions within our university and the one of Hasselt that can help you organize your international affairs. Target these people, all right, and ask them for support. Sometimes you'll have to write for a little application with granting institutions like the Dutch Cancer Foundation, the KWF, or the Royal Academy for Scientific Research in the Netherlands, or any type of fund, and they will usually ask you to adhere to a specific deadline. 2 months in advance, or sometimes they even have very strict deadlines, and they say, okay, in November we want to know. So anyone who has an inkling of, you know, an interest to go abroad should start thinking about that right now and talk to people and organize it in terms of where do I get my funding, what are the deadlines, that sort of thing. All right? So that's what I had to say. And then I have to scoot. I wish you good luck and good science throughout these 2 years. We'll see more of each other. If there's any questions that any one of you has right at this moment, I can try to answer it. If not, the only thing that I wanted to say is that the people who are here from France that are on exchange, are they here? Mallory? You're Mallory? Okay, and you? Somebody said me? Ah, you, sorry, in the nosebleed section. Oh, you're from Portugal. Okay. I have your information, I forgot to take it, in my office. If you want it, come get it. All right? Okay, I'll give the floor to Leo.
Ja, maar zo is het ook.
Oh ja, die had ik ja.
Nee man, die vlag is met groen, die is gewoon met 3 strepen met groen en nog wat. Oh.
Okay, ladies and gentlemen, that was the fun part. I'm a much more serious person than Willem, so this is only serious information now. Mostly on household stuff. I tried to count you and I came up with a number something around 84. That's more than we expected, so I guess a few of you...
Oh my.
Actually, I'm quite happy. I think a few of you weren't there on Thursday, is that right?
Huh?
Yeah, okay. That helps. Carl Otterdijk, I recognize you.
Some of you weren't there.
It's always bad when I know students' names and they've done something before to come to my attention. I will give you some information about a few theoretical things, like problems that you might have the second year. This is just an indication what I'm going to talk about. As you all know, this is a collaboration between the University of Hasselt and the University of Maastricht. You will have lectures on both campuses, which means you have to travel. Most of the Belgian students, I understand, come by their own car, and therefore they need parking here, and that means that they need a UM card. And up to now, Jérôme has 15 of these UM cards, so those people of you that came later will get the card a bit later, but I understand that the Music Spielen will organize that for you. So I hope that by next week we have these cards for you. For the Dutch students, we organize a bus, and Theo will circulate now a list where you put down your name if you want to take that bus. Because if you anyway go by your own car, we'll probably order a smaller bus. But if you intend to take the bus, then please write down your name on that list. Theo will, yeah, that's the passenger list.